Welcome back, ladies and gentlemen, to Aethlon Medical's channel with novel immunotherapies to combat cancer and life-threatening viral infections. Joining us, we have two members of the senior management team to kind of give us a clerical overview of the company through the investor deck. Joining Jim, the Chief Financial Officer, and Guy, the Senior VP. We're going to first and foremost kick it off with Jim. I'll pass the reins over to you.
Thank you, Kyle. And thanks, everyone, for sitting in on our June 2023 update on Aethlon Medical. Here's our forward-looking statement slide. You can read this at your convenience. And you can also find this presentation on our website, corporate website at www.aethlonmedical.com in the investors section. A few highlights on our company. We're focused on combating cancer and infectious diseases with the development of first-in-class immunotherapeutic technologies. Aethlon's led by seasoned industry executives with extensive development and commercialization experience. Our hemopurifier has demonstrated the capture of disease-promoting exosomes and circulating viruses in clinical trials and emergency use. The hemopurifier has been designated as a breakthrough device by the U.S. Food and Drug Administration for 2 indications. We're well capitalized with a solid cash position and no debt. A few key financial highlights. We had approximately $17.5 million in cash as of December 31, 2022. We have no debt on our balance sheet. There are approximately 22.9 million shares outstanding, and our market capitalization as of late May was approximately $7.2 million. We're traded on NASDAQ under the ticker AEMD. Now, I'm going to turn the presentation over to my colleague, Guy Cipriani, to take you through our management team and the rest of the presentation. Thank you.
Thank you, Jim. So we have a pretty seasoned team here at Aethlon, and I'll walk you through a summary of some of the key executives. Dr. Charles Fisher is our CEO. Chuck has had an extensive background in academic medicine as well as industry. He was head of critical care at Cleveland Clinic, and he also worked at Eli Lilly, Abbott, and Cardiome Pharmaceuticals. While at Abbott, Chuck launched Humira, and he's been responsible for the launch of several drugs, including drugs for sepsis. Jim Frakes, whom you've just met, is our CFO and Senior Vice President. Jim has over 30 years of experience as a CFO in public companies. His background includes investment banking and venture capital, and Jim has been responsible for raising in excess of $300 million for the companies he's served. Stephen LaRosa is our Chief Medical Officer. Steve has over 25 years of clinical and research experience in areas of infectious disease, critical care, coagulation, inflammation, and he also has experience with extracorporeal devices. Myself, Guy Cipriani, I have over 20 years of transactional and operational experience with both public and private biotech and medical device companies. Lee Arnold, our Chief Science Officer, has over 30 years of experience in molecularly targeted drug discovery. He's extensively published in peer-reviewed journals and has almost 100 published patents and applications. Our hemopurifier is our core technology. We have an FDA designation for breakthrough device and 2 indications. The first in oncology, the second in treating life-threatening viral infections. To date, we've safely administered 156 hemopurifier sessions in 36 patients. We have a proprietary patented mechanism of action, and we've shown both in man and in vitro that we can clear of life-threatening viruses and tumor-derived exosomes. This graphic simplifies both the mechanism of action and how our device is configured in a patient. As you can see in the upper left-hand portion of this figure, a patient is configured through a catheter and a pumping mechanism to establish an extracorporeal blood circuit, and our device essentially sits in series with that system. Our device essentially can filter the blood by separating the plasma from the whole blood. The fibers in the device are porous, and they have on average, a 200 nanometer diameter pore. So the plasma and these small particles smaller than 200 nanometers, are filtered from the blood, and they come in contact with our proprietary affinity resin. And it's in that resin that we actually capture exosomes and viral particles. This pipeline slide summarizes our core focus right now in cancer and viral infections. In oncology, we're in the planning stages of a solid tumor trial. It's an early feasibility and safety study. And here we're looking at treating patients who are indicated to receive PD-1 antibodies, such as Keytruda, but they failed. And so we're looking to do a safety and feasibility study in those patients. We expect to start that study in the second half of 2023. On the viral infection side, we have an active COVID-19 study that we're recruiting patients for in India. Historically, we've also treated COVID-19 patients in emergency use, and there's some data in some subsequent slides that we can share with you. We've also treated Ebola patients, hepatitis C, viral infection, and HIV. So what is the rationale for our technology in oncology? Essentially, tumors and every cell in the body in particular generate very, very small particles called exosomes. Exosomes are about 50 to 150 nanometers in diameter, and they're extracellular vesicles that are released by all cell types, including tumor cells. Turns out that cancer cells are the most active in terms of trafficking in exosomes, and tumor exosomes specifically have been shown to contribute to the spread of cancer or cancer metastases. They've also been shown to play a role in immune system evasion by the tumor. Tumor exosomes have been shown to facilitate chemotherapy resistance, and they've also been shown to interfere with antibody-based treatments, such as PD-1 antibodies like Keytruda. We believe that the removal of harmful exosomes with our technologies may enhance existing cancer treatments. So in vitro, we've demonstrated that we can remove tumor-derived exosomes with a benchtop version of our technology. So essentially what we've done is, in this benchtop version of the hemopurifier, circulated plasma from patients through hollow fibers and through our affinity resin. And we've shown that in vitro, we can capture about 92 to 99% of the tumor exosomes. We further demonstrated capture from diverse tumor types like head and neck cancer, melanoma, ovarian, esophageal cancer, and breast cancer. So we think we've established a proof of concept in vitro that justifies taking our device forward into clinical studies. We're currently planning an oncology study. This is an early feasibility study, as I mentioned previously. And in this trial, we want to include multiple tumor types, as well as different intervals of treatment, to really establish not only safety, but if we can see signs that our device is working to reactivate things like checkpoint inhibitors like PD-1 antibodies. We're working with a company called NAMSA, who's a global CRO, to help us direct this study in both the US and Australia. And further, we're exploring oncology sites in India for this trial. There's quite a scientific rationale that's developed in the literature recently, which we think provides a justification for our hemopurifier technology in treating severe COVID-19 patients. For example, COVID viremia is detected in about a third of patients, and further, it's associated with disease severity, the requirement for an ICU stay, the development of multiple organ failure, and poor outcomes. Viremia and COVID is associated with immune dysregulation, endothelial injury, coagulopathy, and complement activation. We further believe that exosomes and exosomal microRNAs may play a role in both the spread of the infection, ongoing inflammation, the development of some of the hallmark symptoms of COVID-19 infections like coagulopathy and lung injury. We've also shown in vitro that our proprietary affinity resin can bind all clinically relevant SARS-CoV-2 variants. These data on this slide is from a publication that you'll find in the lower left-hand corner of this slide. And in a severe COVID-19 patient that was treated in an emergency use protocol, we've shown that we can remove harmful exosomes. And in the left panel, these are microRNAs that were contained in exosomes that are associated with coagulopathy or excessive bleeding and clotting in the patient. And we've also shown that we can remove microRNAs which are associated with lung injury. So these data from an emergency use patient, we think help show that removing noxious exosomes may be beneficial to patients. In COVID-19, we're currently enrolling patients in a study in India. And this study has been approved or it's being allowed by the regulatory agencies in India. So the use of our hemopurifier devices in this study is allowed. We're studying ICU patients with severe life-threatening disease. We hope to capture up to 15 patients across 3 centers. To date, we've enrolled and treated 1 patient. The trial remains open, and we've just recently added a second site for this trial. In summary, we believe the hemopurifier can be useful as a clinical tool in both oncology and viral infections. In oncology, we've established the safety and feasibility in a dose-finding study, and we're launching a revised trial that's a safety feasibility and study in both Australia and the United States. In viral infections, we've treated multiple patients, including 2 under an emergency use protocol, and we have an active study that's enrolling patients today in India. We continue to investigate the additional utility of our technology, including in long COVID-19. Long COVID-19, or the post-acute sequelae of COVID-19 infection, PASC, is a condition that is very prevalent in the post-pandemic phase of COVID-19. We have an MTA, or a material transfers agreement, in place with the University of California, San Francisco Medical Center for us to receive plasma samples from them from long COVID patients and for us to evaluate in our technology. In summary, we are developing a novel patented hemopurifier which can purify a patient's blood. And we've actually demonstrated in early trials both viral removal and exosomal clearance. We've been granted by the FDA 2 breakthrough device designations, 1 in oncology and 1 in treating viral infections. We're focused on multiple therapeutic targets, including in cancer and viral disease. We have ongoing and planned US and international clinical trials, including in India and Australia. And we have an experienced management team with a solid cash position and no debt. Thank you for your attention.
And we'll pass the question off to the viewers here. We'd love to know what you think in that comment section below, so we can perhaps do an investor Q&A down the road. And consider subscribing, because as news hits the wire, we're going to continue to update you here. But on that, as always, we look forward to catching you in the next one.